Okay, lots of good answers in the chat. Lanikai, Waialua, Santa Cruz. Fantastic. Let's go ahead and get started. Aloha, welcome. So exciting to see all of you in the Zoom room with us today. We have a great set of talks. My name is Maya Walton, and I serve as the Assistant Director for Research and Fellowships at the University of Hawaii Sea Grant College program. And we are really excited to have you in the audience for the Science by the Sea seminar. In the months of November and December, we're featuring our Sea Grant graduate fellows and our PI-CASC graduate scholars. PI-CASC stands for Pacific Islands Climate Adaptation Science Center. So these graduate students are presenting original research that's supported by Sea Grant and PI-CASC on ocean and coasts. This is research that they're completing while they pursue their graduate degrees. Part of the training that the Sea Grant graduate fellows and the PI-CASC graduate scholars receive is on science communication and outreach. So each of the graduate student presenters that you see today, in addition to conducting this cutting edge research, they are also engaged in community outreach through things like public talks and other activities. So I'm really excited to introduce our three speakers today. And I'm actually going to stop sharing my screen here. So we have three great talks ready for you today. The first talk is going to be from Cassie Ka'apū-Lyons and Emily Conklin. Their talk is titled, "Do you know what baby fishies eat? Neither do we." The second talk will be from Sea Grant graduate fellow Paolo Marra-Biggs, and his talk is titled Plankton, the Invisible Forest. And our third talk today is going to be presented by Honor Booth. Her talk is titled Hāpai e Kawai, Land-Based Pollutants in Kaihuopalaʻai, Puʻuloa. Feel free to enter questions into the chat as we go along. We're going to take one question in between each of the speakers. And whenever you're ready, Cassie and Emily, the floor is yours.
Thank you, Maya. Is it on presenter view or?
Yep, we can see your first slide. It looks really good.
Okay, thank you. Hi, I'm Cassie.
And I'm Emily, and we're going to be talking about baby fish diet today. So our journey starts with a trip to the local pet store to look at aquarium fishes. And we're interested in aquarium fishes because for many people, aquariums are the first and only place they can come face to face with marine species. Roughly 11 million households in the US have a home aquarium, which can help teach people about the natural world. So I wanted to include this quote by author Richard Booth. "We cannot protect something we do not love. We cannot love what we do not know. We cannot know what we do not see and touch and hear." So the exposure that aquariums provide is an important step in getting people interested about protecting marine environments.
As we enter the pet store, we're drawn in by the walls of the colorful fishes. We're first going to take a look at the freshwater fish. The fish we see here are the most popular freshwater aquarium fishes. They include things like plecos, tetras, and fancy guppies. More than 90% of all freshwater fishes in the retail are captive bred. All of the 13 here are captive bred and commonly available in the pet stores, as indicated by the yellow circles. Alternatively, when we look at marine fishes, we again have our 13 most popular aquarium marine fishes, but only 3 are captive bred. Clownfishes, yellow tangs, and clown gobies tend to be common and readily available. The remaining 10 are wild caught. Wild caught means that the fishes have been removed from the reefs. While collections from coral reefs can be done both safely and sustainably, harmful collection methods have been used in the past, triggering both conservationists and environmentalists to respond. As a result, restrictions on collection and export are being put in place. In 2018, Fiji and Indonesia banned coral export, and closer to home, earlier this year in Hawaii, they completely banned collection of ornamental fishes, meaning that the iconic Hawaiian reef fishes are no longer available on the aquarium trade unless they have been captive bred.
So aquaculture of aquarium fish provides a valuable alternative to removing fish from reefs for a few different reasons. One is that captive raised fish have an easier time adapting to tank life and can be bred to be disease-free, and they miss out on the stressful journey from reef to pet store. Another benefit is job opportunities. Culturing marine ornamental fish is a $5 billion opportunity and could provide many jobs. And of course, it reduces fishing pressure on wild stock. But getting fish to breed in captivity is only half the battle. So if we go back to our 13 most popular marine aquarium species, while 10 have actually been successfully bred in captivity, we're only able to produce 3 of these species in commercial numbers due to how difficult the baby fish are to raise.
Baby fish have a long journey before you see them on the reef. After the eggs hatch, marine fishes have a lengthy larval stage. Baby fish are very tiny, smaller than your pinky nail, and can spend weeks to months in the open ocean before finally settling down on the reef they call home. Larvae look very different from their adult counterparts, occupy different habitats, and have very different diets. And in general, very little is known about these early life history stages. Getting fishes through their life cycle in captivity remains the major obstacle in aquaculture industry. High mortality is usually a result of disease and starvation. While disease can be controlled with sanitation and remedial measures, starvation remains more difficult. And this is largely due to our lack of knowledge on what baby fish eat. Because of this, larval nutrition and feed formulation plays an essential role in the aquaculture industry. So we are asking, what do baby fish eat in the wild? Because of their small size and scattered presence in the ocean, it is difficult to observe what larval fish eat in the wild. So instead, we're trying something new. We are sequencing all the DNA information of a whole fish, including what they might have eaten. To do this, fish are collected from the wild using plankton nets. They're taken back into the lab to be sorted, and larvae of interest are individually blended, and the DNA is extracted. The extracted DNA is then put into a machine, which generates millions of copies of target DNA. This is then put into a sequencer, which determines the order of the sequences and puts it into a language that we can interpret. Because we are examining diet and there is potential for fish larvae to eat more than one prey type, multiple sequences are returned and separated by unique barcodes.
So once we get those DNA sequences back, we can do some detective work to figure out which species they came from. So we can look at certain DNA sequences that are unique to a particular organism called barcodes. Just like barcodes link products to prices at the grocery store, these DNA sequences can be linked to the organism they came from. So for example, if we find a particular DNA sequence in the digestive system of this baby fish, we can look at all of the species barcodes we know and match the food item to a particular species, in this case, this little shrimp. And you can do that by using something called a barcode database, which is, again, a lot like the computer used by a grocery store. We use a couple different databases to help look up species, including the Barcode of Life and the NCBI database GenBank.
The benefit of this technology is that it allows us to identify all potential prey items in the stomach of a fish, as well as examine the diets of multiple fishes all at once. We can use plots like this to help us visualize the data. The fish species is on the left and the prey type on the right. Plots like these allow us to better visualize what baby fish are eating and whether different species have different diets. Do baby fish have broader diets like the species in red seems to indicate, or are they more picky and feed on just a few prey types like the species in dark blue? We can also see that some food types are more popular than others and are eaten by a lot of different species.
So our goal for this project is to apply this advanced genetic technology to raising baby fish. Once we're able to identify what larval fish eat in the wild, we can set the table for aquaculture. We can work with growers to identify nutritional requirements for popular species that have been difficult to grow in captivity. This has the potential to greatly diversify and enhance the aquaculture industry.
We aim to help push aquaculture into the future. Not every potential prey item is currently culturable and inexpensive, but with results from our work, we hope to make it far easier to raise these prize fish so that reefs and aquariums can both feature the beauty of marine ornamental fishes. And with that, we would like to thank all those who have put time and support behind this project and our funding sources. Thank you for listening.
Thank you, Cassie and Emily, for your talk. Applause from the audience. I can see the clapping hands emoji going up on various Zoom squares. We'll take questions from the audience now. If you have a question, please send it to us via the chat. And while we wait for those questions to roll in, I'll ask you guys a question. Have the results about what fish eat, has anything been surprising to you?
Yes. We found that a lot of fish are eating krill, and they do have an aquaculture facility that is breeding krill in Japan, I believe. And normally everybody feeds baby fish copepods. That's the assumed diet.
Thanks, Cassie. I see one more question in the chat right now from Nancy. Nancy, would you like to ask your question out loud?
Okay, I was wondering if this information from your study has been put to any practical use in the tropical fish raising industry that you know about? Thanks.
Hi, Nancy. Thank you for your question. Not yet. This is pretty new, and we're still working through the data.
Thank you.
Great. Sounds like those conversations are coming up next, which is fantastic. I'm going to bring, thank you again, Cassie and Emily, for your talk. I'm going to bring Paolo up to the Zoom stage now.
Hello, can everyone see my first slide?
We can, Paolo.
All right, well, thank you for coming today. My talk actually pairs quite well with Cassie and Emily's. I'll be speaking about plankton, which we will learn is a basis of a lot of the food chain in the marine ecosystem. So hopefully you will walk away with a newfound appreciation of this microscopic life. And while this is not directly related to my current research, as you'll see, it does influence my research subject as it does all beings in the ocean. So to start, I want you guys to take a deep breath. For every other breath we take, we can thank plankton. They are the invisible forests, in particular, single-celled photosynthetic phytoplankton in the world's oceans. And remarkably, these elegant microscopic cells perform a bit more than half of the photosynthesis and oxygen production on Earth, equal to all the land plants combined. And some of these organisms help turn a CO2-dominated volcanic Earth into the livable, oxygen-rich environment we have today. But even so, they remain largely invisible to the vast majority of the world. But who are they? They're more than a biological class of organisms. Plankton are rather an ecological or behavioral niche. Plankton, the word, comes from the Greek word planktos, which means wandering or drifting. And any living creature whose own propulsion is less than that of the currents is classified as plankton. So you swimming in a gushing river mouth technically are plankton. They're a diverse group in shape, color, and size, and they range from the tiniest virus to siphonophores, which are a relative of jellies. They are the longest animals in the world. But to define them a bit further, to break it down, there are two distinctions between the types of plankton. There's phytoplankton, or plant-like plankton, and zooplankton, or animal. There are also some bacterioplankton, fungus, and mycoplankton, but they make up a small percentage by weight. And we love the oceans for the menagerie of charismatic, majestic, and diverse animals. And all the animals that we see, whales, fish, manta, seals, they only comprise 2% of the ocean's living biomass. The remaining 98% consist of mostly invisible multitudes of plankton. And there's one specific kind of phytoplankton called Prothorococcus that I would like to highlight here. There's an estimated 3 billion of these organisms in the ocean. And in one single drop, there can be up to 20,000 individuals, with an estimated global mass equal to 220 million BW beetles. And this organism is responsible for 20% of the oxygen in the atmosphere alone. And what's a fun fact, we only discovered this species in 1988. And so there are so many plankton in the ocean that if you added them all together, they'd outweigh every single animal by far. And to define phytoplankton a bit more, there are thousands of species, around 5,000 species of known photosynthetic phytoplankton. And main categories, main types of organisms are diatoms, cyanobacteria, dinoflagellates, and coccolithophores. We owe what's not to love? We owe every other breath produced by these phytoplankton. They are the largest carbon sink, and they intake dissolved CO2 from the seawater. Some species emit light called bioluminescence and make beautiful scenes in the water. And last but not least, and most important, they're incredibly crucial for all of life in the marine ecosystem. They're the ocean's buffet, and these tiny phytoplankton comprise 70% of the ocean's biomass. Phytoplankton that you can't even see with the naked eye. And they are the foundation of the marine food web, of which all of our favorite charismatic megafauna rely. And fun fact, just like in every food chain, we can trace the photosynthetic organisms to the basis. We can also see them in our landscapes. Cliffs of Dover in Southeast England are made up of phytoplankton largely. They're made up of coccolithophores that I mentioned earlier, a type of phytoplankton that looks like this with a chalk-like skeleton. And over 100 million years ago, these cliffs were underwater, slowly building and slowly accreting until they got uplifted. And we see them today. But it's not just in the cold water like England that we have plankton. Even in these clear tropical waters, coral reef animals depend on plankton in the water. Most of these animals in this photo, including the giant clam, rely on plankton because corals and clams adopt the phytoplankton and use it to their advantage in their tissues, like thousands of mini solar panels in their skin. And there are many projects at HIMD right now that are investigating the symbiotic relationship between animal and phytoplankton. And in temperate and cold polar places, there are much longer hours of summer daylight. They have stronger seasonal flux of phytoplankton. And this is an 8-year time lapse of the Earth breathing. And you can see the phytoplankton boom and bust as the winter and corresponding summer seasons pass. And the general trend is the further away from the equator you go, the more biologically rich the oceans get. The water's colder, and there it can contain more oxygen in the water, and there's more nutrient mixing, as well as longer daylights for the phytoplankton to survive. And all these factors are important in creating ideal conditions for phytoplankton production. So they're small organisms, but they can have large impacts, both ecologically and geologically. However, you can have too much of a good thing. Most algal blooms are natural and beneficial, usually signifying the most productive seasons of the year, the summer and fall. However, a balance is needed. When there's an excess of nutrients, you can have sometimes detrimental effects, such as harmful algal blooms, or HABs, as they're known. They're caused by an overabundance of nutrients, specifically nitrogen and phosphorus from terrestrial inputs. And some of these phytoplankton create a neurotoxin as a byproduct that when they're in such abundance, they can be very harmful to the animals that ingest them. And then there are also non-toxic harmful algal blooms that can create oxygen bed zones from the decomposition and the algae depleting the ocean, the oxygen in the ocean. Just like bog areas, these dead zones can create anoxic or anaerobic conditions and not allowing many fish or zooplankton to live. And what keeps them in check? What keeps all this phytoplankton in check? They're predators, the zooplankton. And they're the early developmental stages of the animals that you may think of living in the marine environment. The sponges, the crabs, the sea stars, the snails, worms, and some fish. And to avoid these predators, many zooplankton hide out in the dark twilight zone beneath the photic zone in the ocean, beneath the depth of water where sun can penetrate. But every day, they make a huge migration to feed on phytoplankton that live above in the photic zone. And during dusk, they move up in the water column and make their way down during dawn, which causes a migratory domino effect, making it the largest daily migration in the animal kingdom. And this happens every day. And to highlight one species of zooplankton, which Cassie mentioned earlier, the copepod, or the ocean cocoa puffs, because you need to eat thousands to be full, are the most abundant multicellular animal in the oceans, or even on the whole planet. There's an estimated 1 quintillion, that's 18 zeros. And zooplankton are quite diverse, and they can go through many changes throughout their life. Some you wouldn't be able to recognize, and they may look drastically different from each other. Think of a caterpillar and a butterfly. So this is a zoe larvae that turns into yet another planktonic stage before its final recognizable form. And often these stages will eat different things as they grow, sometimes phytoplankton, sometimes each other. And this creepy critter called phronema is a voracious predator. It actually kills gelatinous plankton and uses the hollow corpse as a home to lay eggs. Sound familiar? Because it's the animal that inspired the horrific creature in the Aliens movies. See the resemblance? So the world is filled with complex and alien life that go through strange changes. Similar to how we have, in the womb, we have a tail and webbed feet before we're born. But not only do they go through wild transformations, they also inspire science fiction stories. Zooplankton are incredibly important as they truly are the sustaining factor in all of the marine life as we know it. And to end this talk, I'd like to quote John Steinbeck in his log from Sierra Cortez: "The disappearance of plankton, although the components are microscopic, would probably in a short time eliminate every living thing in the sea and change the whole of man's life." And it's true. We rely on them, on phytoplankton and zooplankton in so many ways that you wouldn't be able to expect. They supply us with every other breath. They regulate the climate. They're the base of the food chain. Heck, plankton even made it into many of our toothpastes. So with that, I'd like to say thank you to Sea Grant, to everyone who came and heard this talk, and to plankton itself.
Thank you, Paulo, for your talk. Great to learn about plankton and the role that they play in our oceans and on our Earth. If there are questions from the audience, please drop them into the chat. We're happy to take a question for Paulo at this point. And while we wait for those questions to roll in, Paulo, can you tell us a little bit about the inspiration behind this talk? I know this is something outside of your dissertation research. What inspired you to tell us about plankton today?
Well, I'm originally from California, which is one of those cold, temperate regions that have a lot of plankton. You can see it in the water, and most people think that plankton don't exist, or most, people are surprised when they hear that plankton exists in tropical waters. And I study an organism that relies upon plankton to survive. They get, just like corals, they get about 90% of their energy from photosynthesis, from these phytoplankton that they put in their tissue. So I thought it was a fun subject to talk about. There's so much to know, and they're widely overlooked. Just to think that all the phytoplankton in the ocean outweigh all the animals. That's pretty amazing of a fact that I think we should learn about and really appreciate phytoplankton more.
Thanks. I'm going to be thinking about plankton and phytoplankton the next time I'm out in the ocean, and I think you're right, they are overlooked. We have a question in the chat from Dr. Brian Bowen. Brian, would you like to ask your question out loud?
Sure. Nice talk there, Paulo. Do plankton occur in fresh water, and do they matter?
They do, very much so. Yeah. So they just because their environment is less salty doesn't make them any less important. They are still, as you can think about it, the food pyramid, everything that we rely on, all the fish, all the animals that we use for subsistence, they depend on something beneath them. And at the basis of the food chain, we have the phytoplankton that live in fresh water, and all the larvae of all the animals that we are used to, the zooplankton, depend on them. Good question. And
Thanks.
I see one more.
Yeah, let's take this question from Dr. Beth Lenz. Beth, do you want to ask your question out loud?
Sure, thank you. Great talk. So out of curiosity, knowing how much we rely on plankton and communities, how sensitive or robust are these plankton populations, and how might that sensitivity or robustness differ by the region within the ocean or freshwater systems?
Good question. So I did find some research indicating that there has been a decrease in phytoplankton abundance along with warming oceans, because I mentioned they are very susceptible or sensitive to the amount of nutrients in the water. And the idea of a warmer ocean decreases the amount of dissolved atmospheric air that can be in the waters, of which phytoplankton will use for their metabolism, for their photosynthesis. And so there is a decline in phytoplankton, and there were some projects trying to raise phytoplankton to actually offset some, or to start some carbon sequestration, which is kind of a large project to imagine, but they are robust enough because of their ability to reproduce. And let's see, the second half of the question, do you know if this differs by regions in the ocean? Yeah, so I think I showed in the presentation a time lapse of the seasonality of the oceans, seasonality of the world, and you can see in certain regions there are far less chlorophyll concentrations, and that's in the tropical regions away from coastlines. And you get the strongest mixing, the strongest nutrient upbringing from deeper water near coastlines and in temperate and polar regions. I hope that answered your question, Beth.
Thank you.
Thanks, Paulo, again for your talk. Another round of applause. And we're going to bring our next speaker, Honor, to the Zoom stage. Honor, we can see your first slide. It looks really good.
Okay, good. All right, aloha mai kako. My name is Honor Booth, and I'm a graduate student in the Department of Chemistry at UH Manoa, working with Dr. Megan Donahue, Dr. Eileen Nally, and Dr. Kim Felinsky at the Hawaii Institute of Marine Biology. Our project, Hāpai Kawai, Carried by the Water, investigates land-based pollutants in coastal ecosystems, especially in Kaihuopalaʻai or West Loch of Puʻuloa. Looking at the transmission of pollutants to local communities through seafood. So today I will talk about the purpose of our project a little bit and give some background information about our study site, specifically land use and restoration efforts. I'll then share some preliminary results and next steps for our project. So as many of you may know, Hawaii's geography has deep land and sea connections, and human activity on land affects the health of coastal ecosystems. This is true when the ʻahupua'a system was thriving and has not changed despite the changes in land use that we see today. The goal of our project is to better understand how pollutants from modern human development are transported by streams and waterways to coastal ecosystems, and in some cases, back to the community through fish consumption. The breadth of the ewa moku allows for different modern day land uses, including modern and historical agricultural use at the base of the Waiʻanae Range and further down in the Ewa Plains, residential and industrial uses along the Waikele Stream, and recreational uses such as golf courses along the coast. Due to the different land uses in the ewa moku, we expect to find a diverse set of known pollutants. Right now, we are focusing on Kaihuopalaʻai or West Loch, our study site in Puʻuloa or Pearl Harbor on Oahu. Kaihuopalaʻai is subject to stream inputs from 3 ahupua'a in the ewa moku, Honouliuli, Hoʻaiʻai, and Waikele. We have partnered with 2 restoration projects that are responsible for our target collection sites. Pohala Marsh, which is under the responsibility of the Department of Land and Natural Resources, Division of Forestry and Wildlife, and Kapapapuhi Point, which is under the responsibility of the nonprofit Mālama Puʻuloa. In Kaihuopalaʻai, we were able to collect sediment, fish, and crabs from for pollutant analysis. So far, we have results back from for sediments collected from the south of Kapapapuhi Point Park, as well as, so this is Honouliuli Stream here. We collected sediments from north of Honouliuli Stream, the mouth, as well as south of the stream mouth. And then we also collected sediment samples from the mouth of the East Fork of Waikele Stream, as well as sediments from a bridge on Kapakahi Stream, and from the mouth of Kapakahi Stream. All the sediments were analyzed for heavy metals such as aluminum, arsenic, chromium, and lead, polyaromatic hydrocarbons such as naphthalene, pyrene, and anthracene, polychlorinated biphenyls, which have been banned since the 1970s, and organic chlorine pesticides such as DDT, DDE, and DDD, which have also been banned since the 1970s. To evaluate the levels of contamination, we used NOAA's Threshold Effect Limit, or TEL, and Probable Effect Limit, or PEL, for freshwater and marine sediments. TEL is the sediment contamination concentration at which a toxic response has started to be observed in the benthic organisms. And the PEL is a sediment contamination concentration at which a large percentage of the benthic population shows a toxic response. All preliminary results that I'm going to be sharing with you are organized. So the figures will all have the, the TEL and PEL values for freshwater sediments, marine sediments, or if the values are the same, they'll be denoted with the black dotted or solid lines here. So you can use this legend here to follow along. I would also like to add that we analyzed sediments for organophosphate pesticides. However, these compounds were not found in any of our sediment samples. And then just to remind you about the different sample sites as I go through the results, I will show the map again. So we're going to get started with the results for Kapapapuhi South, which is this area here. As you can see, no organochlorine pesticides or polychlorinated biphenyls were found in the sediments here. However, we did find heavy metals and polyaromatic hydrocarbons. None of the polyaromatic hydrocarbon concentrations were over any of the PEL or TEL values, except for this one compound here, dibenz AAH anthracene, which only exceeded the TEL for sediments. For the heavy metals, you can see that the maybe heavy metals of most concern would be arsenic and chromium, as these values do reach or exceed both the TEL and the TEL values for freshwater and marine sediments. The next site we'll be looking at results for is the Honolulu North area. So again, no organochlorine pesticides or PCBs were found here. We did see an increase in the concentrations for polyaromatic hydrocarbons, which suggests that the sediment here is collecting more than moving. And then also we do see a slight increase in some of the heavy metals. The next site that we're going to be looking at is the Honolulu Mouth site. You can see here that compared to north of Honolulu Mouth, polyaromatic hydrocarbon concentrations are much lower, and the heavy metal concentrations are slightly lower. The arsenic is one to point out that over here, the values are lower than the TEL for marine and freshwater sediments. The next site we'll be looking at is Honolulu South. So this is south of the mouth. Again, we see a decrease in polyaromatic hydrocarbons here, as well as the heavy metals. And again, one of the heavy metals that might be concerning with the arsenic is also, again, lower here. So now we're going to go across the lock to Pohala Marsh and look at results for sediments from the East Fork of the Waikele Stream, collected here. So here you see that we see organochlorine pesticides here, but no polychlorinated biphenyls. We did find DDE, which is the breakdown product of DDT, and very long-lived in all environments pretty much. And then we also see two other organochlorine pesticides, endrin ketone and heptachloroepoxide. The value for heptachloroepoxide does exceed the PEL for both marine and freshwater sediments, which is incredibly concerning because there's actually a lot of people who live along this stream, like fishing camps, and they regularly eat or sell their catch from the stream. And you can see polyaromatic hydrocarbons are a little bit higher than some of the sediments from across the way. However, none of them except for the benzo-A anthracene and again the benz-AH anthracene exceed any of the thresholds. Oh, and the phenanthrene also exceeds one of the thresholds. And then you can see the heavy metals. Here, chromium exceeds all of the PEL and TEL values for both types of sediment. As we go over to Kapakahi Stream, things get a little bit scarier. We're going to look at the Kapakahi Bridge sediment results first. Here we see all of our pollutant groups. The PCBs, here we see one of the congeners. There is DDE, which is present and higher than let's see, the PEL threshold for freshwater, as well as both of the TEL thresholds. All of the polyaromatic hydrocarbon values are incredibly elevated here as well. And then some of the heavy metals exceed the TEL values for both types of sediment. And you can see all of the heavy metals that we analyzed for in this sediment as well. Next, we're gonna go downstream to the mouth of Kapakahi Stream. Again, we see all of our pollutant groups here. Two types of PCBs, DDE and DDT. This is really interesting because DDT breaks down very readily in the environment to DDE. So seeing DDT here, thinking that it's been banned since the 70s means that there's like a fresh source of it close by, and is probably is likely the source for the DDE. Again, we see all of the heavy metals and polyaromatic hydrocarbons. The polyaromatic hydrocarbons concentrations are incredibly elevated compared to the other sediments that we analyzed. But I think that for all of these, the presence of the DDE and the DDT is really concerning. Pohala Marsh is, it's a restoration site for a lot of native birds, and DDT and DDE are known to cause the shells of birds to become thinner and crack. So if DOFA is trying to restore this area for native birds, removing the sediments from this area that have those chemicals is really important to encouraging the bird survival. So some next steps for our project is we still have more samples to be analyzed, sediments from Waikele Stream Bridge and the west fork of the mouth, also Honolulu Bridge and Nihola Fish Pond. I'll just show you really quick. Nihola Fish Pond is this triangular shape here, a little bit north of Honolulu Stream. It's actually, it's not a settling pond for the stormwater. It's really interesting in there. It's very closed off to any other water sources except for rain, king tides, so exchange from the ocean during king tides. And then other than that, the storm drain feeds it. So I'm kind of excited to see what we find in there. And then we also have fish from Honolulu Stream mouth and Waikele Stream mouth, as well as samoan crabs from Waikele Stream. And then of course, we're going to continue to work with our community partners to provide baseline data for moving forward with their restoration projects and any outreach that they are doing to let the community know about the hazards of eating seafood from these areas. So with that, I'd like to say mahalo piha, especially to our community projects. Auntie Sandy and Ati at Malama Puʻuloa and Evan are super valuable to our project and have helped so much with sample collection and access, as well as our partners at DOFA and DLNR, allowing us to access the areas so that we can get our samples. And then also to all of you for listening. And with that, I'd like to say, hey, ma'aninau ka oko. Do you all have any questions?
Thank you, Honor, for your talk. Round of applause for Honor. Let me add the spotlight for her. What happened? Here we go. Oh, there we go.
Just sharing.
There we go. Great. I do see questions coming into the chat. A round of applause for you. Thank you so much for your talk today. Let's take Paulo's question first, and then we'll move to Brian's question second. Paulo, would you like to ask your question out loud?
Yeah, I was just wondering if you could use these sediment analyses as some sort of a forensic tool to investigate where the fresh source of DDT is coming from.
Yeah, so there's actually, I should have mentioned this. There's a dump site. There's a couple dump sites close to Pohala Marsh. The actual area, I'll actually here, let me share my screen again so you can see the map. So this area right here is actually filled in dump site. And let's see, so if the bridge is here, yeah, so this is all a filled in dump site area. And then this is actually where the marsh is, where the water and the birds are. And then this is another dump site here. So, and this I think ties into Dr. Bowen's question as well, is that this is probably where the DDT is coming from, as well as the PCBs, is this dump site. I'm not sure how old it is, but I know that it's capped off, and when you look at it, you can see they have the little the tubes for the volatiles to gas off. It's, yeah, I never want to go in that area again, quite frankly. But I think that would be the closest thing that you could do for forensics. Maybe, if you could ask the city and county to get some sediment cores from the dump site area, and then trace it back. But yeah. And then let's see, Dr. Bowen's question, why is Kapakahi so much more polluted? Yes, so again, that's because of the proximity to these dump sites. Can it affect human health? I'm sure if you were eating the seafood from this area long enough, especially the benthic organisms versus the fish can move and swim, and they're in the water column. A lot of these organic compounds, while the water does move them or move the sediment which carries them, they don't want to be in the water so much because they're more I'm gonna get chemistry here, a little bit more hydrophobic. So if they're going to get trapped in the sediment, I would think if you're eating crabs from there, you're at higher risk than if you're eating fish from there. Although I've personally wouldn't eat anything from this area.
Thanks, Honor. What I'd like to do now is I'm going to bring our other speakers back to the Zoom stage, and we'll have an extended Q&A session now. So if you have questions for any of our speakers, please put your questions into the chat. And while we wait for those questions to roll in, I'll ask all of the speakers, what's one piece of information that you hope the audience walks away with today that you want them to share with friends, families, coworkers, their neighbors? What's one thing that you hope people walk away from your talks with today? And we'll start with Cassie and Emily first, and then we'll cycle through the talks.
I guess one thing from ours is that a lot of fish are just way more complex than people realize, and that the fish you see on reefs start off as little tiny babies that look like completely different than their adults, and basically, yeah, live in different habitats, eat different foods, and I think that's just sort of a cool, complex part of the ocean that, yeah, is fun to learn about. Cassie,
Yeah, to add on to that, larval fish matter and they're difficult to rear.
Thank you. I like that message. Larval fish matter. Let's move to Paulo. What's one thing you want us to take away from your talk today?
Yeah, I hope that people appreciate phytoplankton a bit more after hearing some of these facts, and value the health of our oceans as they do the health of our rainforests, and both are being mistreated. So hopefully everyone is re-inspired to be stewards of the land and water.
Thanks, Paulo, for that message. And Honor, what's one thing you want us to think about, walk away from your talk with today?
Oh my gosh, don't eat any fish from Puʻuloa, pretty much. Don't eat any seafood from there. You know, while I'm doing my field work out there, I'll like get lunch and people see like my paddleboard or like how muddy I am, and it's like pretty obvious I've been in the swamp. And I tell people what I'm doing and they get excited, like want to buy my samples from me so they can eat them. And I'm just like, no, don't do, like, please don't eat these. So yeah, spread the word.
Thanks, Honor. Okay, so we have larval fish matter, appreciate phytoplankton, don't eat fish from Puʻuloa. Thank you for those messages. I do see a question in the chat. Beth, would you like to ask your question out loud?
Of course, Maya. Thank you for the opportunity. Cassie and Emily, this question is for you. Based on what you're finding from your baby fish gut content, how specialized or generalized are their diets? And how does this differ maybe by genus or species? I don't know how deep in the level you get.
That's a good question, Beth, and one we're still working on.
That's great.
This data is very preliminary, and so we're still working through another set of larval fish to kind of be able to better answer that question.
Yep. Yeah, I guess to add on, like super preliminary, but it does seem to depend a lot on which kind of baby fish we're looking at. But great question.
All right, I don't see any more questions in the chat, so another round of applause for all of our speakers. Thank you for your excellent talks today. Oh, there's a question that just came in.
Sorry.
Beth, would you like to ask your question?
Yeah, if we have time and if Honor doesn't mind. I had a question in terms of your research, like that's really important data, as you were saying, especially like if you're eating or gathering from the area. So how often have you been able to, or are there plans to engage and connect with local residents in that area or users in that area to help them better understand water resources? There is the Clean Water Act, but in terms of just, this data is coming in and this is what we're finding, and just empowering communities.
Yeah, so Malama Puʻuloa, they actually have an outreach kind of like portion of their project. So we are, how should I say this, like our results are going to be used by them for their outreach campaign that they're doing in the community, since they they work with the community a lot closer. And I like I said, talking to people while I'm doing my field work, when I access the streams at the parks or something like that, people know, like they, when I tell them like, oh, we're testing the sediment or we're testing like the crabs and stuff, they get really excited and they ask like, oh, did you find anything? Like they know about the dangers, but also there's that like, well, like, I don't know, maybe emotional eating. I don't like wanting to eat what you want to eat, kind of thing. And people kind of like, or maybe wanting to continue to, fish the way that, their families have for years. So it's that balance of like, okay, if we tell the community members, like, you shouldn't fish here, where are they going to go to fish? And then like, how does that impact other communities, who are also doing the same thing? And I think that like the frequency at which we can let people know, like this isn't a monitoring project, unfortunately, simply because like there's no lab here on the island to be able to process these samples. Hopefully when I'm done with school that can change, but like there's no way to be able to like update people really on how things are changing because of how prohibitive analyses for these things are here in the islands. And I think, I hate to mention, like, I think the Red Hill thing is really bringing that to light for like everyone, realizing that like we need a lab here on island that can do all of these services, because our resources are being impacted just as much as they are in the continent. Yeah, I hope that answers your question. I guess
Definitely.
often,
Thank you.
not very often.
Well, that was great. Thank you, Honor.
Thanks, Honor, also for highlighting that need to bring these water quality analyses to Oahu and to Hawaii. Thanks for mentioning that. So that's all we have today for the seminar. Another round of applause for our speakers. Please join us next Thursday. We'll have a set of 3 new talks at noon on December 16th. And thanks for coming today. Thanks, everyone. Aloha.